Okay, so if this means that Chapter 2, which is where we're picking up for the Bio 211 final exam review, is where we just finished off, you should be able to describe the 4 functional groups of organic compounds, who's saturated, who's unsaturated. That would be fats, right? Levels of structural organization of a protein, primary, secondary, tertiary, quaternary. Notice some enzymes, I would say, all enzymes are proteins, but not all proteins are enzymes. Enzymes require coenzymes to be most efficient, but you'll notice enzymes and catalysts do the same thing. Enzymes are organic catalysts. Okay, so if we're going for complexity, now we're going to move on to here, where here is Chapter 3. Okay, because now we're going to take all of those basic structures, and we're going to build a cell. Okay, you should be able to understand the structure and the function of a typical cell membrane. All of the organelles are made up of membranes. What's happening there? Remember, we are creating lipid bilayers that trap specific chemical reactions inside of specific spaces. If you think about, say, the mitochondria, we make ATP there. Okay? We make the largest number of ATP molecules there because we trap all those chemical reactions in that space. What's the difference between passive and active transport? Well, passive transport would be, say, diffusion or osmosis. Active transport is that sodium potassium ATPase you're still having nightmares about. Sodium potassium ATPase, okay, that's used to generate an electrochemical gradient across the surface of essentially all of the cells in the body, right? If we have cells, what happens to them when we put them inside different environments? And what do we mean by that? The example that I obviously want you to think about is going to be what happens to our red blood cell. So let's add some color to your life. Our red blood cell. What happens to the red blood cell? Notice I'm giving you a specific shape to the red blood cell. It's a biconcave disc, no nucleus. Essentially is transporting hemoglobin, proteins inside here, and the hemoglobin molecules bind to oxygen. So if it is an isotonic solution, it is normal. So let's just put N here. Okay, if it's in a hypertonic solution, what happens? Make sure you understand the difference. Hypotonic solution, what happens? Okay, it means that there's some amount of salt outside here and some amount of salt inside here, and water is going to move to dilute that solute, and water is the solvent. The 2 major sequence questions that students have a difficult time with, okay, for the final exam have always been events in protein synthesis. And that means you have to go from DNA to RNA to protein, right? And even that requires a little bit more in the way of explanation. DNA to RNA requires a specific enzyme. What is the enzyme? RNA to protein requires the processing of that RNA, so you end up with mRNA. And that mRNA is going to be made into primary strands of amino acids that fold on top of themselves, secondary structure, and then finally its functional shape is its tertiary structure. Now I know I made this look pretty straightforward, and it is really that straightforward. Go to the figure inside your textbook, scramble the images, maybe draw them on flashcards, mix them up, put them back in the correct order. Same thing with cell replication here. All cells grow. All cells grow to a specific size. Once they grow to a specific size, we replicate all of the DNA. So you go from 23P to 46P, 46 pair, okay? And then we take that one cell and we make 2 different cells by undergoing mitosis, right? So you're going to make 2 smaller cells. Okay, that should be about the same size as we started with over here. The significance is this, right? We've duplicated the entire genome, and then we have to go through prophase, metaphase, anaphase, telophase, right? And all we're doing there is we are separating the duplicated strands of DNA from one another. So you actually have now 23 pair here, 23P, and 23P here. So hopefully this different way of looking at it helps you understand it a little bit better. So it's chapter 3. Now think about this. We now have populations of cells that we can use to build tissues, and tissues are going to be used to build organs, and organs have to be put together to build organ systems. And remember, you're made up of 11 organ systems that are required for you to live. So what we're doing is we're increasing complexity. Let's get rid of these cartoons, change the slide, and to increase complexity, we move to tissues. There are 4 basic tissue types. I promise you, you have to know the 4 basic tissue types. You have epithelia, you have connective tissue, you have muscular tissue, and you have nervous system tissue. And by nervous system, N-E-R, okay, we mean the excitable neuron, muscular tissue, the excitable muscle cell. Epithelia, connective tissue, non-excitable. In fact, why don't we do that? We just go like this. I'm going to go like this. I'm going to say no, and we're going to go plus over here, where these are excitable, muscular and nervous are excitable. Group them together. Epithelia and connective tissue are not excitable. Okay? That's not to say that muscular tissue doesn't have connective tissue with it, right? That endo, peri, and epimysium all make up tendons and ligaments, right? And those tendons and ligaments are connective tissue. It's not muscular tissue. It's not the excitable tissue, right? Likewise, nervous system tissue. If we're building a blood-brain barrier, those have microglia, they have ependymal cells, they have oligodendrocytes, These are not excitable cells. They are all connective tissue. Okay? Now, it says here, know the locations and functions of all tissues outlined in class. That's a lot of tissues. But how did we do it? Remember what we did is we put tissue here, and we put these guys E over here. We probably put connective tissue over here. And then we put, I don't know, muscle and nervous system over here. We put a little arrow over here to group them together as excitable. And then remember what I did? I went little arrows like this to here, where we went from solid to liquid. And that means liquid connective tissue has specific cells floating around inside of it. RBCs, liquid connective tissue. White blood cells, I don't know, neutrophil, NEU. Remember, they're in the little cartoon. You definitely listed them inside your lab handout. Those are all connective tissue. Epithelia, remember, they come in 2 basic types. They're either simple or they're stratified. And they're simple squamous, cuboidal, columnar, modified columnar, right? Muscle, we have 3 different types of muscle cells. You have cardiac, you have smooth, notice I'm not drawing any striations, and you have skeletal muscle tissue. Right. Now you've, obviously the neuron we studied the most this semester was the multipolar neuron, and you know where we're going to head with that, right? And here's a way to remind yourself of this. Let's imagine this is in the central nervous system. That means these cells here are oligodendrocytes. And remember, oligodendrocytes are connective tissue. Okay? But let's think about like the epithelial cells. Let's put 2 or 3 of these guys next to one another. And these are all basically cuboidal cells. Right? And these cuboidal cells are sitting on top of a basement membrane. And then how do we hold them together? Okay, know the locations of cell type junctions. And, oh my God, what an annoying question. I think start off with know that there are cell junctions. What's the difference between an adherens, gap, so on and so forth, right? But there's a pattern. There is. Go from the most upper to the lowest, right? So desmosomes are here. Hemidesmosomes are here. Tight junctions are on top. Gap junctions allow ions to pass through. Okay. And then if we have epithelial cells, we can build glands. Remember? Epithelial cells are going to build glands. Glands are going to be important when we get to, say, chapter 15, right? Because we're going to innervate those glands with sympathetic or parasympathetic stimulation. So the glands unicellular, multicellular. If they're either of these, are they myricrine, apocrine, and holocrine? Which one is the most common? What I'd do if I were you, I'd just learn the definitions of these. Okay? You know they're made up of epithelial cells. Okay? Cool. So now we're starting to increase this complexity. Let's get rid of the cartoons. Wham. And let's add some more to it. Chapter 5 represents, now that we actually have tissues, we can build a system. And that system, of course, is the integumentary system. Okay? And the integumentary system is describe the structure and function of skin and its exocrine glands. So that means go back to the drawings you made for skin inside your lab packet. Differentiate between thick and thin skin. Lucidum. Okay. Who has hair? Who doesn't have hair? Okay, nor the order of the epidermal strata and what cells are inside each strata. If I just drew this quickly for you, right? It'd be like, hey, strata. Corneum on top. Lucidum if it's thick. What's below it after that? You know, this is the basal, so the place most people are going to make mistakes are going to be here. So granulosum or spinosum first. Which order? Make sure if you're reading these questions inside your exam, know the order. Well, if we're starting outside here, it's a specific order. If we're starting inside here and we're moving this way, it's a different order, right? The significance of that is, how would you apply that information to, say, phases of deep wound healing? Okay? It's an important concept to understand that we need to keep that cutaneous tissue as whole as possible. And to do that, when you actually cut yourself, cause cells to grow from the bottom up. Okay? Yes, no, maybe. And in doing so, they first have to fulfill the rules of contact inhibition down here. But there's a phase to it, right? There's a bleeding phase. There's a clotting phase. There's a callus removal phase. And then finally, you actually have the replacement of the tissue itself. But of everything inside Chapter 5, think about what you have to do first. You have to know the structure and the function of skin. Differentiate between the two types inside the body. Know the order of the strata inside all of that skin, and then what happens when you break it. Okay? So let's get rid of those, move on to a little bit more complexity, so that we're ready for something like this. And I think what I did is I jumped directly to 12, right? What is excitable and what is not excitable? Neurons are excitable. And if there are neuroglial cells, what are the types of neuroglial cells? What do ependymal cells do? How do they function? Okay, and you know that these are not excitable, so don't get fooled by this. At the end of the day, though, really Chapter 12 is about building an excitable system. And for the sake of simplicity, what I'm going to do is I'm just going to draw like this one. And notice I'm intentionally drawing more than 2 neurons, right? So that you can work through each of the different steps. Now, I'm not putting any myelin on these guys, so these are all interneurons, right? Which means they're C-type fibers, right? Okay? But that means if we're going to release an excitatory neurotransmitter to bind a receptor here, that means we're going to have a series of events that will be summed at the axon hillock here. Right? And if it reaches a specific value, we then have an action potential that travels along here to release the next neurotransmitter. And let's just say for yucks and giggles that it's an inhibitory neurotransmitter. So that would mean you would actually cause something like that. What is that representing? What doesn't happen here, and therefore what will never happen here. Okay? So that's what we want you to be thinking about. And notice I put 3 neurons here, so you can sort of fight your way through this review part. I can stand here and draw what happens. Yay, summation, depolarization, all of that. And you need to know this. Do not get me wrong. This is going to be a sequence question. Okay, we're going to take this, we're going to break it apart into smaller pieces, not literally the cartoon. We're going to break it apart into smaller pieces, and you're going to have to reassemble it properly. Okay, what is happening here? Where are those sodium ions moving? Where are potassium ions moving? How do we get back to minus 70 millivolts? That was supposed to be a 7. Sorry about that. 70 millivolts, right? MV. Okay. Whoa, something's happening to the drawing here. Okay. So, If we went back to the beginning, you'd see that, we want you to think about each of the different sections inside this. What I'm going to do is I'm going to pause here and I'm going to finish up with the next 2 or 3 slides and spend some time developing the complexity of how you should be thinking about this. And notice this is as complex as I need you to do if you understand everything else that's going on here. But maybe you don't. Maybe what you have to do is you have to draw these 3 neurons out. And maybe you have to literally just copy what I just wrote out for you here. And then maybe change it. Imagine this is an interneuron that activates a myelinated neuron. And how does that change our story? If this is myelinated, it could be an A-type fiber or a B-type fiber. Okay? I didn't do this for simplicity. I did this to make sure the picture didn't get too busy. Okay. For now, that'll do. Thank you for your help and attention.